Hi all, today let's learn about cell foundations and advanced microscopy. Okay, so in biology, cells are recognized as the fundamental units of life, right? Every living organism, from single celled bacteria to complex multicellular organism, is built from cells. So understanding cells has been one of the greatest achievements in science, made possible through the development of increasingly powerful microscopes. Early scientists would only observe basic cellular structures, but modern microscopy techniques now allow us to visualize organelles, proteins, and even molecular complexes at near atomic resolution. So here we will begin with the principles of cell theory, explore essential microscopy skills, and examine advanced techniques for such as electron microscopy, freeze fracture, cryogenic electron microscopy, fluorescence microscopy, and immunofluorescence. Together, these technologies reveal the intricate architecture of life. Okay, so let's begin. So, cell theory forms the foundation of modern biology and consists of three major principles. What are they? First, all living organisms are composed of one or more cells. So, first, we have to check the composition, right? Whether an organism is microscopic or enormous, cells are built-in blocks. Okay, so no matter whether it is big or small, all the all these kind of organisms are built up of different cells, right? Secondly, the cells are the basic structure and functional units of life, right? So what are their functions? So every biological process, including metabolism, growth, and reproduction, occurs within the cells. So we have designated cells for that. Thirdly, the origin. All cells arise from pre-existing cell through cell division. So this principle explains how organisms grow, repair tissue, and reproduce, right? So cell theory was developed through an inductive reasoning where scientists observed many living organisms and identified common cellular characteristics. Okay, so once established, the theory could be applied deductively to predict that any newly discovered organism would also consist of cells. So this relationship between observation, prediction, and this highlights the scientific method in action. Okay, so the specific observation of all life is cellular. Okay, this is what the theory says. Clear? So cell theory forms the foundation of modern biology, explaining structure, growth, and disease. You can see a microscope here and some parts. We have a coarse adjustment knob which help to move stage a large distance. Fast rough focusing to locate the specimen, right? And then we have a fine adjustment knob, move stage a small distance, slow precise focusing for a sharp image. Okay, so before advanced microscoping can occur, scientists must master microscope operation and specimen preparation, right? So for microscope, it contains two important focusing mechanisms, as we mentioned. The coarse adjustment knob moves the stage a large distance and is used for rough focusing. The fine adjustment knob moves the stage a much smaller distance, allow precise image sharpening. Okay, and now let's see how to prepare the sample. So preparing a temporary slide involves several careful steps. First, a thin selection of tissue is placed onto a clean slide. Okay. So the select section part. Okay. Then a drop of water or stain is added to improve visibility. Okay, we have to hydrate the tissue. Then a cover slip is then lowered gently to prevent air bubbles from forming. So we need to put a cover. Why? We need to remove the air bubbles. Okay. Finally, excess liquid is removed to produce a clean specimen. So we will press it in order to remove the excess liquid. Proper specimen preparation is essential because even the most advanced microscope cannot produce quality image from a poorly prepared sample. So both are equally important. Clear? Now let's see the calibration and the measurement framework. So microscopy is not only about viewing structures but also about measuring them accurately, right? An eyepiece graticule is a small ruler placed inside the microscope eyepiece. However, its scale has no fixed unit and must be calibrated. Okay. So calibration problem. The calibration is achieved using a stage micrometer which contains a precisely measured scale. By comparing the two scales, scientists determine the actual dimensions presented by each graticule division. Okay, so calibration assigns physical unit to arbitrary graticule line, ensuring reliable scientific observation. Then the magnification. How to calculate magnification? Magnification calculations are also essential. The relationship can be expressed as image size equal to actual size multiplied by magnification. Okay, clear? So using the formula, scientists can determine the size of cells, organelles, and other microscopic structures with accuracy. So quantitative measurements transform microscopy from single observation into a powerful scientific tool. Clear? This is how we transform the data. Now let's see how to do the mapping. So microscopic technique exists a long spectrum of increasing resolution. So at the lower resolution end are the light microscopy techniques, including fluorescence and immunofluorescence microscopy, right? So these are particularly useful for studying living cells and tracking biological process. Okay, so first one is light microscopy, so on. Clear? So moving towards higher resolution, we encounter electron microscopy. Okay, techniques such as scanning electron, right? Freezing fracture, microscopy, etc. Okay, this is a transition zone. Then at the highest resolution level are the transmission electron microscopy and cryogenic electron microscopy, which can reveal structures at the angstrom scale. Okay, so this is the finest. So this progression demonstrates how scientists choose microscopy methods depending on the level of detail required. So different techniques answer different biological questions. Clear? Electron microscopy uses beams of electrons rather than visible light, providing much greater resolution. So the electron microscope. Scanning electron microscopy or SEM examines the external surface of specimens. Samples are coated with the metal and scanned by electrons. Okay, so here we have the method. Reflected electrons generate a highly detailed three-dimensional image of the specimen's surface. So here we will get a 3D image. Okay. So what are the applications? We can see the insects, pollen grains, external cell surfaces, etc. Right. Then we have transmission electron microscopy or TEM. It focuses on internal structures. So their extremely thin sections are prepared, allowing electron to pass through the specimen. So the resulting image reveals organelles and the internal cellular architecture. Okay. So the method is here. So SEM is ideal for studying morphology and surface features. Okay. While TEM excels at visualing internal ultrastructure. So it provides a 2D image of the internal ultrastructure. What are the applications? We can study about mitochondria, ribosomes, nucleus, etc. Okay, so together they provide a complementary perspective on biological specimens. Okay. Freeze fracture microscopy is a specialized electron microscopy technique used to investigate biological membrane. So the specimen is rapidly frozen to preserve its structure. It is then fractured using a super cold blade. Okay. Because the membrane represents weak points within the cell. So the fracture often passes through the lipid bilayer. After fracturing, ice is removed through a process called etching. Okay. A thin platinum carbon replica is deposited onto the fractured surface. The biological material is removed, leaving behind a detailed replica that can be examined using a electron microscope. Okay. So the freeze fracture provides unique insight into membrane organization, revealing embedded proteins, membrane topology, and protein distribution across cellular membrane. So this is how the structure will be visible. Okay, so we will get an exact 3D distribution. Now let's check the cryogenic electron microscopy. Okay, this is cryo. Cryogenic electron microscopy, commonly known as Cryo-EM, is one of the most powerful imaging techniques available today. Okay. Samples are rapidly frozen using liquid nitrogen or liquid ethane. Okay. Preserving them in a near natural state without chemical fixation. So maintaining specimens at extremely low temperature reduces radiation damage while allowing the scientists to expose them to higher electron doses. Okay. So this significantly improves the image quality and resolution. So cryo-EM can reveal molecular structures at the angstrom level details and have revolutionized the structural biology. Scientists now use cryo-EM to determine the structures of proteins, viruses, and molecular machines that were previously impossible to visualize. Okay, so we have to check the resolution. It is in the angstrom level, right? Then preparation rapidly frozen in liquid nitrogen or ethane. And what was the native state? Specimen remained hydrated, preserving natural biological structure. Protection, it has significantly reduced the electron beam radiation damage, so the damages are minimized. What are the applications? The definitive tool for isolated proteins and the viruses, right? Now let's see the fluorescence microscopy. So fluorescence microscopy allows scientists to observe dynamic biological processes in living cells. So this technique relies on fluorescent molecules called fluorophores, which illuminated by specific wavelength of light. Okay, so the major advantage is observing living cell in real time. Okay, a common example is green fluorescent protein or GFP. Okay, the GFP. By attaching GFP to a protein of interest, scientists can monitor the protein's location and the movement inside the living cell. Okay. So unlike electron microscopy, fluorescence microscopy is particularly valuable for studying processes that changes over time, such as cell division, protein transport, and gene expression. Clear? The immunofluorescence microscopy combines the specificity of antibodies with the visualization power of fluorescence. In the primary antibody method, a fluorescent label is attached directly to an antibody that binds to a target protein. Okay. In the secondary antibody method, an unlabeled primary antibody first binds to the protein. A fluorescently labeled secondary antibody then binds to the primary antibody, amplifying the signal. So this is how the secondary antibody method is working. So because antibodies recognize specific protein, immunofluorescence allow scientists to identify the precise location of molecules within the cells and tissues. This technique is widely used in medical diagnosis, cancer research, and infectious disease investigations. Okay, so these are very useful out there. Now we have the microscopy selection matrix. Choosing the correct microscopy technique requires balancing resolution and biological application. So fluorescence and immunofluorescence microscopy provide lower resolution but allow visualization of living cells and specific proteins. Scanning electron microscopy and freeze fracture microscopy offer nanometer scale resolution and detailed structural information. Transmission electron microscopy and cryo-EM achieve the highest resolutions, relieving molecular and atomic level structures. This selection matrix highlights the trade-off between observing dynamic biological processes and obtaining the highest structural detail. Scientists select the method that best address their research question. Okay. So in short, cell theory established that the cells are basic unit of living organisms, and all these microscopy has transformed our understanding about life. All these technologies create a continuum of discovery, allowing scientists to move from studying entire organism to investigating individual proteins as microscopy continues to evolve. So it will provide even deeper insight into the structure and functions of life itself. Thanks for listening.